अंकुर जी, can you see?
यस
हाँ हाँ
सर।
सर, यस सर।
सर एक बार प्लीज मूव करिए स्लाइड, लाइक फर्स्ट सेकंड मूव हो रहा है नहीं हो रहा। मूविंग सर, मूविंग मूविंग, ग्रेट।
Okay,
यस सर।
good.
डन सर।
Only slide दिखा रहा है ना आपको?
यस
जी
सर,
सर।
यस।
Okay, ठीक.
थैंक यू।
Thank you.
रुचि जी, ऋचा शर्मा जी,
Yes, sir.
हाँ आप प्लीज प्रोसीड करिए।
Sure, sir. Hello, everyone. This side, Ms. Richa Sharma, Program Coordinator of Nature 2024. I welcome you all on 30 days of International Winter School, Navigating Agricultural Transformation for Upliftment or Rural Excellence, Nature 2024. Today is 14th day of a training organized by AgriMeet Foundation, Bharat, in collaboration with CMET Mexico, Southern Federal University, Russia, ICARDA, Lebanon, World Agroforestry Center for International Forest Research, Kenya. ICRISAT Hyderabad, Manage Hyderabad, SKLT, SHU Telangana, MGUVV Durg, CCSH AU Hisar, IGKV Raipur, IRI New Delhi, MHU Karnal, NDRI Karnal, ICR IRI New Delhi, ICR DCR Puttur, Karnataka, Stanford University and University of California, etc. So we have Dr. Prakash Ganga Shetty. Dr. Prakash Ganga Shetty ji is working as senior scientist in ICRISAT, Hyderabad, India. He is leading with pigeon pea breeding program at ICRISAT, Patancheru. His research focuses on developing early maturing, climate resilient, disease resistant, pest tolerant, and nutritionally rich pigeon pea varieties and hybrids of Asia and Africa. He has developed the world's first speed breeding protocol in pigeon pea and are currently deploying with molecular marker assisted selection to accelerate product delivery and enhance genetic gain. Speed breeding enhanced the cultivar delivery process in 5 years compared to 15 years for the traditional method of developing new varieties and hybrid for dryland worldwide. He is recipient of Monsanto's B. Shell Bullock International Scholarship for PhD program in genetics and plant breeding from the University of Agricultural Sciences, Dharwad, India, and University of Wales, United Kingdom. Also worked as postdoctoral scientist at University of Wales from Feb 2013 to March 2014. He moved to ICRISAT as PDF Pearl Millet Breeding in April 2014. He led Africa's Pearl Millet Improvement Program from October 2016 to January 2021. His major achievements have been the identification and utilization of the first Striga-resistant line in pearl millet, the development and successful release of Africa's first FA biofortified millet variety, Chakti, and hybrid ICMH 147007 in ECOWAS, development of field-based rapid generation advancement for pearl millet in West Africa. He has published more than 55 journal articles with citation exceeding 350. He had been a leading principal investigator for BMGF-funded Steiger control in pearl millet and sorghum. Harvest First Challenge Biofortification Program for West Africa. United Kingdom Innovates funded a project on delivering low GI pearl millet to control type 2 diabetes in the African population, and SML Lab of the Feed the Future funded a project on genetic enhancement of pearl millet for yield, biotic and abiotic stress tolerance in West Africa. He has received several awards, including the Bayer Ted Crosby Vishal Bolong Impact Award in 2020, the Promising Young Scientist Award in 2018 for Outstanding Pearl Millet Breeding Work in West Africa, and the Resource Mobilizer Award 2019. He has guided one PDF, four PhD scholars, and master's students. So the session is yours, sir. Over to you.
Thank you, Richa ji. Ankur ji, shall I proceed? What is the procedure now?
Yes, sir. Please proceed, sir.
Okay. Thank you. If any questions or anything, if I'm not audible, just let me know. Okay.
यस सर. लेक्चर के बाद सर 15 मिनट का डाउट क्लियरिंग सेशन रखते हैं. चैट बॉक्स में पार्टिसिपेंट्स अपनी क्वेरी को लिखते हैं, हमारे मॉडरेटर आप तक क्वेश्चन पहुंचाते हैं सर, आप उसका जवाब देते हैं.
Okay. Thank you. No problem. Okay. So good evening, everybody. Thank you, Dr. Ankur, for inviting me to give this presentation on innovative approaches in pigeon pea breeding to deliver higher genetic gain. As Dr. Richa already given a brief introduction about me. So I was working as a millet breeder in West Africa, based in Niger. So currently I'm leading the pigeon pea breeding program for Asia and also Eastern Southern Africa, mainly working in India, Bangladesh, Myanmar, and Kenya, Tanzania, Malawi, and Mozambique. Especially these are the countries where pigeon pea grows. So this is my topic, wherein we will be dealing with the innovative approaches in pigeon pea breeding to deliver higher genetic gains. So as we know, what is pigeon pea actually? So if I take pigeon pea as a crop, it can give us 4 important major aspects for as a person or as from belonging to this country. It is one of the most climate resilient crop, and because of its sensitivity and thermosensitivity and also multiple resistant to some of the diseases, it makes one of the crop where it can adapt to wherever you don't get much water actually. Then it also contribute to food security, because pigeon pea is the one crop where India we eat a lot of pigeon pea, at least 3 times a day. Then it also gives a nutritional security aspects, particularly which is high in protein content, then also in high calcium, magnesium, and folate content as well. Then it also has a high TSS and very bold grain, which can also be used as a vegetable, and also you can use as a dal. Then apart from that, after food security and nutritional security, it has a major contribution toward the soil health, where it can contribute to the biological nitrogen fixation and the nitrogen use efficiency. Then because of its deep-rooted system, it can also have the high carbon capture from the atmosphere and fixes into deeper layers of the soil. So this can also become one of the crop where you can go for the carbon capture from the atmosphere and reduce the climate change issues associated in the nature. So this is one of the most resilient crop wherein it can contribute to the major these 4 portions. When we come to its importance as a crop, specifically it is adapted to the rain-fed farming system, and it gives a multifaceted soil benefits, as I told in my previous slide. And it also has a health and nutritional benefits. Apart from protein, it can also give a source of vital elements for bodybuilding. When we come to crop cultivation status in India, specifically India is the one where 70-80% of the production comes, and globally it grows in around 6.3 million hectares. And India itself, it will produce around 4.4 million hectares, with a production of around 5.47 million ton globally, and in India we are around 3.6 million ton. And average productivity is almost same in globally and India as well, around 860 to 824 to 860, maximum around 50 30-40 kg difference between globally. The major things associated with is the production is around 3.67 million tons in India, but the requirement is almost the double than what we grow currently in India because of its production constraints. So totally worth of around 633.83 million USD, we import pigeon pea from different countries to India. So with respect to Atmanirbhar Bharat, we need to produce and meet our local demand and also reduce the importation of pigeon pea. So we need to increase the area production productivity and make some innovative approaches in the current breeding program to achieve this requirement and make the self-sufficiency in pigeon pea production in India. When we took the scope of this crop in India, specifically there are more than 79 new varieties released in last 10 years from the AICRP and also from state agriculture universities. So if you take the overall growth of this crop in 2010, we produced around, the requirement was around 3.5 million tons, but production was around 2.46 million tons. And if you go to 2030, the projection is around 6 million tons has to be produced. But the current scenario is around 5.5 million tons we can achieve. But the requirement, again, it is a double than what we are projecting here. So it needs always an importance toward this crop to meet that particular domestic demand of the crop. And also there is a MSP, higher MSP for this crop. That is a minimum support price from the government, around 7,550. Now the major challenge is the area is stable, but the productivity is not that much great. So we need to increase the area as well as the production and productivity. Up to March 2024, we imported almost around 1.2 million tons of pigeon pea from different regions of the world. When we come to ICRISAT, actually, so ICRISAT is the global center for breeding for pigeon pea, and mainly started our breeding program in last 1972. That is almost a 55 years old pigeon pea breeding program at ICRISAT, and which is the one which caters the needs and demands of global pigeon pea germplasm requirement and also the production productivity. For example, if you take the major milestones, what ICRISAT done in last 50 years is that in 1986, ICRISAT brought the first Pygiarium wilt resistant variety, that is Maruti. Until today, it grows in the farmers field almost around 20-30% of the area covers because of its durable resistance to Pygiarium wilt in the farmer field condition. And in 1991, first time in the world, which ICRISAT brought the pigeon pea hybrid, that is ICPH8, that is the GMS hybrid. And any hybrid in the pulse crop is the first is having pigeon pea. And in 1993, ICRISAT again brought one mega variety that is called Asha. Many of you know about this one. This is the first wilt and assembly resistant variety brought into the cultivation by ICRISAT's breeding program. And in 2004, ICRISAT brought the Hybrid Parent Research Consortium formed with the private seed industries to deliver the pigeon pea hybrids to the farmers field. And the major breakthrough happened in 2008. ICRISAT bought the first commercial pigeon CMS hybrid into the commercial cultivation. And in 2011, pigeon pea genome sequencing has been done. And in 2017, 300 lines sequenced. And 2018, a high density array has been developed for genotyping purposes and also for the trait mapping. And in 2021, import genome assembly has been published in public. And in 2023, ICRISAT and Indian Institute of Pulse Research Kanpur, and with the help of state agriculture universities in 13 states, we launched a project to promote the early maturing pigeon pea varieties in India with the funding support from Department of Agriculture and Cooperation, Government of India. Then in 2024, we brought the world's first pigeon pea speed breeding protocol published, and which is the first major milestone in any of the crop which is having highly photoperiod and thermosensitive, where we captured its protocol and published, and currently we are using in the breeding program. The importance of this and those things will be explained in my next slides. So at global level, in ICRISAT, around 19 countries, 135 varieties released in different countries, and 2 commercial hybrid released from the ICRISAT bred material up to date. And in ICRISAT also in collaboration with India, Myanmar, Eastern Southern Africa, Australia and South America. There are around several varieties which are released in Myanmar, and currently they are growing in the Myanmar region, and they are the one who actually produces and export to India as an export potential crop from the Myanmar. And apart from that, ICRISAT hosts also the Global Pigeon Pea Field Days for the partners, then produces integrated approaches for pod borer and pod fly management, and identification of maintainers from African germplasm and capacity building of the NAS programs globally. So when this is about the introduction of what ICRISAT has been done in last 55 years on this crop, and with the recent modification in our breeding programs, we are bringing new market segment into the crop. Specifically, when we do a market segmentation, that means for which particular purpose we use this crop, and how their breeding can support to these market segments. So here specifically with the first market segment, we'll be working on high quality split grain, that is dal varieties for the market segmentation 1. And that caters to the Asian program, specifically India, Nepal, Bangladesh, Sri Lanka and Myanmar. And wherein we put around 70% of our breeding effort to scatter, because around more than 70% of the pigeon pea comes from this particular area around the world. So major effort has been dedicated to the developing high quality split grain or the dal varieties. Then second market segment we work on is the vegetable pigeon pea and whole grain consumption. That is called as market segment 2. Specifically, this is harvested when the crop is very young, and we take out that one and process it, then feed into the market. And this particular market segment scatters the needs of Eastern Southern Africa, especially Kenya, Tanzania, Malawi, Mozambique, Ethiopia, and Zambia, wherein the vegetable pigeon pea is grown and it is consumed as a whole during its greener stage. And at ICRISAT, we'll be putting around 30% of effort for this particular market segment to breed the varieties which are vegetable purpose. So overall, it is 70% is the grain type, that is a dal purpose kind of varieties, and around 30% is for the vegetable pigeon peas. So what are the major challenges in pigeon pea production actually? So when we have the rainy season production, so recently there is something called as Phytophthora blight because of the heavy rain during the cropping period that has caused the waterlogging and also the Phytophthora. And after once the rain stops, because of heavy rain, the dry root, because of the dry spells, it also attacks the dry root rod. Then we have the pod borer, which is the major challenge to nurture the pulses, and here at pigeon pea as well. And Fusarium wilt and Stolti mosaic are the major challenges. And we have now immune lines which are resistant to both the diseases. Then also we have the pod fly. And in abiotic stresses, we have the waterlogging. That is the major challenge for the currently pigeon pea production. And drought and heat often causes also a major yield loss to the crop. When you go to the, because the pigeon pea, you know, most of us, the pigeon pea is produced in the rainy season. And now, because of its demand and also to increase the pigeon pea production and productivity globally, so we are pushing the crop to the post-rainy and summer season. But when you go to the post-rainy and summer season, major challenge is the photoperiod sensitivity and agronomic dwarfing. That means if you start growing the pigeon pea in different months of the year, it will show different reaction to its plant height. That means the plant height is directly correlated to its day length. So that is the agronomic dwarfing we say actually. So we have to identify a proper stage where pigeon pea can grow at the and get a proper yield. Then also we are seeing the seed shriveling is a major challenge where we are getting the shriveling of the seeds. Then cold tolerance, if we are pushing the pigeon pea in post-rainy season, specifically the early stages of the crop growth is reduced by its cold. So we need to have the genotype which are also having the tolerance to cold condition in early phase. Then we have the heat tolerance. Again, if you grow for summer conditions, the flowering stage heat tolerance must be incorporated into the lines which you are bringing in the summer. So overall, when we come to the crop, there are several breeding challenges associated with this crop, specifically the generation turnover time. Because of its highly photosensitive nature and also thermosensitive nature, the crop is restricted to only one season per year. And as we know, the genetic gain is proportionally correlated with the time taken to produce that particular line. So that's why the generation turnover, if you have only one, then the genetic gain or the varieties which comes into the farmer's fields takes a lot of time. Almost in pigeon pea, it takes around 12 to 13 years to bring a particular line to the farmer's field. So that's the problem with this crop. So that's why we need the speed breeding and other protocol developed for this crop to take at least 3 to 4 generations per year to improve its genetic gain nature. And also it has a photo and thermosensitivity that need to be taken out from the crop, because photo and thermosensitivity restrict the crop to take only one season. And if you don't have, if you have the photo thermo insensitive material, that can be grown throughout the year. And agronomic dwarfing, as I explained in the earlier slide. Then another thing is pigeon pea is a perennial crop. As long as it's got water, it will continuously grow actually. So that also need to be kept because the farmer can take a particular variety for particular duration. Then after that, he can go for a second crop if the moisture availability is there. But in many of the cases, it's not happen because of its longer duration and also because of its perenniality. So annuality nature should be brought into the crop. That will also explain what is that annuality we are taking care of in pigeon pea breeding. Then challenges of emerging disease like Phytophthora and row rot also causes a major yield loss to this crop. Then abiotic stresses like waterlogging, heat, cold, and drought also causes sometimes because the crop duration itself is more, so that it passes all the 3 stages of its life cycle in these 3 environments. Then seed swelling in post-rainy and summer. Sorry. Then herbicide resistance because because of its sluggish nature of early growth, the weeds competition is very high. So you need to have a herbicide resistance in the crop where it can take care of that one. Then plant types or mechanical harvesting because of the labor situation now. We need the plant which can be harvested mechanically. Then under investment and research, although we are telling Atmanirbhar Bharat has to be brought in this crop, we need to have a good amount of investment and research to bring such kind of material into the farmer's field. So now, for example, as now we discuss what are those major challenges, then how to improve its pigeon pea production and productivity. For that, Government of India has brought 2 particular kind of interventions to improve its production and productivity. Especially, there's 2, we call it as a vertical expansion and the horizontal expansion of the pigeon pea. So what is that horizontal expansion is means that development and promotion of short and early maturing disease-resistant, climate-resistant varieties, then popularization of good agriculture practices, then advanced seed plan, that is the distribution of promotion of high-yielding varieties which are in the advanced stage of testing, then improved farm implements and processing units to reduce the post-harvest losses, and also capacity building through technology transfer and increase the seed rate, that is SRR around 45% and varietal replacement as quick as possible. Then also improve the productivity per unit area of the crop. So this is called vertical expansion, and currently at ICRISAT with the NAS partners, we are implementing this particular kind of strategy in the farmer's field. Then in horizontal expansion means bringing the non-traditional area into the non-traditional areas and low productivity districts into the pigeon pea production area, and also moving the crop to post-rainy and summer condition. So bringing such kind of genotypes also we are doing at ICRISAT, and pigeon pea newly completed big irrigation projects, then pigeon pea in the rice fallows. So all these needs a special attention of the researchers to breed and to bring new material for the horizontal expansion. But vertical expansion, already we have the material that we are putting onto it. So after this particularly vertical expansion, horizontal expansion, what is the most important thing which is important for the cropping is product profile development. For example, if you take India, we have many states within India, and every state has its own weather conditions and also the soil and geographical locations. We need to have a proper product profile development for the each category of lines which you are going to take. And based on this product profile, the breeding efforts can be put. So for that, it is that we started in 2016 for modernizing our breeding programs to enhance the genetic gains. We done a workshop with all the national program partner. And also last year we conducted a global pigeon pea field day from different stakeholders who can be guiding the pigeon pea breeding programs around the world, what is the requirement and how we can proceed. So with this, what are our objectives of breeding? Especially we'll be dealing with 5 major objectives that like delivering climate resilient, disease and pest tolerant high-yielding varieties, high-yielding especially dal varieties for South Asian farmers and consumers, and also breeding for medium duration climate resilient pigeon pea varieties with the dal purpose. Then breeding for mid early and early duration. And we also have the super early duration, which matures in 90 days, and breeding for the hybrid pigeon pea parental lines and hybrid for the early duration group. So these are the 5 major breeding objectives of our pigeon pea breeding program. So for that, we have developed target product profiles, wherein you can see first in we have 4 target product profiles for pigeon pea. In first target product profiles, we'll be dealing with the medium duration climate resilient varieties with resistance to disease and tolerance to pests. And majorly the percent effort, if I take 100% of my effort as a breeder, so we are putting for this particular target product profile around 25% of our effort for 3 million hectares. And target states like central and southern zones of India, especially Maharashtra state, Karnataka, Telangana, AP, MP, Gujarat, UP, Odisha, and Tamil Nadu. And also it can cater the needs of Myanmar and Nepal. And some of the material which adapt to this region can also spill over countries like Kenya, Tanzania, Malawi, Mozambique, and in some of the Sudan, Zimbabwe countries. So for this developing the target product profile, we also have the product development goals. That product development goals, the line which we breed, it should have these particular traits. Those we called as a must-have trait. And these traits will be achieved within the breeding program of around 3 to 5 years. For example, yield in multi-location yield trial condition, it should be more than 10% over the commercial check, which is already available in the farmer's field. Then we need to have the Fusarium wilt and Stolti mosaic disease virus, the resistance, that mortality percentage less than around 10%. Then maturity of around 160 to 180 days because of its medium duration. Then we also need the 100 seed weight of around 9 to 12 gram test weight. So these are the 5 must-have traits should be captured in our breeding program. Then in long-term goals, like more than 5 years, what we capture is we identify some of the donors for waterlogging, phytophthora, dry root rot, pod borer, pod ply, heat, drought, herbicide, mechanical harvesting, and annuality. So these are the traits we are slowly bringing into our breeding program and streamlining them. So now currently whatever the line comes out of our breeding program, we'll have these 5 must-have traits, and some of the have nice to have traits with the recent changes in our breeding program. So some of these to check the yield comparison with the lines, we are using this newly released varieties like TTRG 59, LRG 33, and GRG 811 as a checks to beat our current breeding programs. And in second breeding pipeline, specifically we deal with the mid early to early maturity. For example, this is the one which take care of the early segment actually. And that also goes to major parties like central and southern zones of India, and spill over again to those now countries like Eastern Southern Africa. Here you can see the earlier in medium duration, we have 5 must-have traits, and in early maturity, we have 6 must-have traits. The one added to this one is the photoinsensitivity. Because of if we need earliness, we need to have the photoinsensitive, and also we are bringing the thermoinsensitivity so that the breeding lines can be moved to different seasons, and where you can get the higher yield advantage compared to other seasons. And also we can expand the pigeon pea area into the non-traditional areas of post-rainy and summer conditions. And in the long-term goals, more than 5 years, again the same traits what we are looking for here. And here again we have the checks like UPAS 120 and IPA 15-6. And one more segment we are bringing is the super early pigeon pea varieties. Super early is nothing but again the varieties which can mature in less than 90 days. So if we take the normal crop duration is more than 6 to 8 months, but the particular market segment or the target product profile deals with the varieties which can mature in just 3 months. That means half of the what current varieties are there. So again, we have 6 must-have traits where also we are looking for seed weight of 9 to 12 gram, maturity of 90 to 100 days. And here complete photoperiod insensitivity material we are looking for this particular market segment. So already we have a release variety that is Puja Hara 16 from Puja Hara 16 that we are using as a check for this particular market segment. Then the last, the target product profile is we are having the hybrid pigeon pea program. And in the world, this is the only breeding program where we have the pulse hybrid that is in pigeon pea. And currently Ecrisat is the one which is looking for this hybrid pigeon pea in early maturity durations. So here also we have 5 must-have traits. Again, the hybrid in early duration and the yield advantage has to be more than 15% because this is a heterosis yield advantage. Then again, we have WSMD maturity and 100 seed weight are within the limit of what we have designated for the early maturing pigeon pea varieties. So here one important thing is that when you talk about the hybrid, we have the female parent program, and also we have the male parent program. And female and male parents are different, so that I will explain them in my further slides. What are those states we are looking into the male and female? So when we come to the AICRP zones and our target product profile, AICRP zones, like we have 5 AICRP zones in India, that is Northeast Plain Zone, Northwest Plain Zone, North Hill Zone, Central Zone, and Southern Zones of India. So these are designated from the AICRP based in IAPR Kanpur. And area coverage within those zones and also our target product profiles. For example, if we take the South Zone, we are working on all the 3 target product profile. That means the testing of these lines coming from those target product profiles will be tested in these particular zones. Like that, we'll be testing in different target zones with the different target product profiles. So overall, when we come to the Pigeon Pea breeding program, so as I told, this is the we are doing only mainly on the dal purpose varieties, and also for the vegetable pigeon pea if it goes to the Eastern Southern Africa. So we have 4 market segments, and especially for 3 markets, we do 3 target product profiles we do with the varietal group, and for the 4th target product profile, we do a breeding. And in all the target product profiles, we have yield, maturity, weight, SMD, and 100 seed weight, particularly appropriate to those particular duration will be taken care during the process. And in hybrid pigeon pea program, we have the seed parents and the restorer program. So those 2 different breeders will be looking after for the developing the seed parents and the restorer programs. So when we come to these target product profiles, how we breed a particular variety to that particular target product profile. For example, if we are breeding for particularly for the target product profile 1, and that target product profile 1 will be looked after by the breeding, that is breeding pipeline 1. That means a particular, this entire is a 1 breeding pipeline for the 1 target product profile. If I am breeding for a medium maturity variety, so you can see how to bring a particular, for example, you might have studied already the breeding schemes in our college days and in MSc and PhD. So this is the commercial breeding scheme which applied to the pigeon pea breeding in our current breeding pipelines. So you see the year here, then seasons, how many seasons we take per year, then breeding pipeline, how many number of lines, male and female we use, then when we phenotype particular material, then when we use the genomics like the trait markers, and those we'll use in this one. So since this breeding pipeline deals with the medium duration varieties, we need the photosensitive condition because all medium duration having highly photosensitivity in nature. So wherein we take the 15 crosses by selecting the allied parent from that one. And the allied parent is based on yield and some of the allied traits and its adaptability to the particular target ecologies. And that will be, the lines will be selected based on the mid-density panel based dissimilarity index, so that you will not be selecting the similar ones to cross. So we are using dissimilar lines which are alike in nature and with the different traits to introduce, combine to a particular genotype. That will be done in the season 1, and season 2 we do the F1s around 15 crosses, and those F1s will be confirmed through quality check markers for hybrid confirmation. Then from the S1, from the F2 to F3, we will be going with the single seed design method and the rapid generation advancement. That rapid generation advancement will go in a particular chamber that I will take in the next slides. So around from 15 courses, we do around 4,500 plants, and from 4,500 plants, we go for single plant spacing, and we select a particular material. And also some of them, if anybody needs a bulk of F4, and also we share with the NARS. Here mainly we see for flowering, maturity, then pods per plant, then 100 seed weight. Those are the must-have traits what I mentioned in our target product profile. Those we take care of selecting at the F4. From 4,500 plants, we take around 600 plants in the rainy season. And those 600 plants, we again grow them in a design with augmented design with the checks. There we select for yield, flowering, maturity, HSW, SMD, and FW. And simultaneously, we also screen the 600 material for FW and SMD in the SMD-SIG plot. That will also show in my further slides. wherein we will select around from 600 progenies to around 600 lines to around 50 progeny lines here. And those 50 progeny lines will go for the multi-location yield trials across the India. And from that multi-location yield trial, we select from 50 to 10. And those 10 again will go for the stage 2 trialing in the across India again. From here, if we identify at least around 3 to 4 best lines, and those will be nominated to the AICRP trialing, that is IVT1, AVT1 and AVT2. Like that, if any material which is found to be highly good, then the NAS program take that one and nominate to the AICRP program. Like this, we breed a particular variety for the particular market segment. So here for the second market segment, for example, you can see in first market segment, we are doing only 15 process here. We are restricting the time and also bringing a lightness in the parents and also bringing a new material within the group. In medium maturity, with the use of speed breeding, we are almost ending a life cycle of particular line in around almost around year 3 or year 5, because once we have the stage 1 testing and identify best 4 to 5 land, those will go for again as a light and use them in the crossing program before bringing into the new material. So when we come to the major market segment of around bringing early maturing pigeon varieties, this is the major breeding effort we are putting, because this is the what requirement of the current farmer needs actually. So here you can see here we are ending a life cycle of variety within 3 years. There in medium maturing varieties, we are ending life cycle of a particular product. That means bringing line within 5 years. But here, it will take just 3 years to bring a line here. So we'll be doing around 40 crosses here. Again, with the meat density panel, dissimilarity index, and the quality check markers to check whether the true F1 will be there or no. From each 40 crosses, we use around 12,000 plants, around 300 plants per cross. Then those F2, F3, F4 will all grow them. Up to F3, F4 will go in the RGA. From F4 onwards, we'll go single plant selection. Mainly, we choose to select them for photopid insensitivity. That too by using, you see the year. The first season will be coming under the F3 of RGA. Then second, it will go to summer selection. From summer, it will take call out around, we'll select only around 10 to 20%. From rainy season, we will take out some around around 10 to 20%. From this year 2, we'll be passing actually rainy, post-rainy and summer. And whatever the line you taken during that time, we'll be saying whether it is photopid sensitive or insensitive or thermo insensitive or sensitive, because it will be passing all the summer, rainy and post-rainy condition. Then once we have around 250 lines selected from the FI material from the end season, and this will also go simultaneously for wilt and SMD. We'll check for all the must-have traits from this 2,000 progeny, then we select around 250. And with these 250s will be seed increased for the multi-environmental testing. And those will go for more than around 70 to 80 locations across India for multi-location testing. That is we call it as a stage 1 testing. From this 250, we will take another around 10 to 20% of the selection. Then we go to the 20. We select around 20 progeny plants, 20 lines, which will go to the stage 2 testing, and also anybody interested, they can go for the nomination to the AICRP system. Ankur ji.
जी सर.
Yeah, I think Uma Shankar has raised a hand. We can take the questions now or we can take at the end.
सर वो हो जाता है, बाद में ही रखते हैं सर, लेक्चर के बाद में ही डाउट क्लियरिंग सेशन रखते हैं. मैं पार्टिसिपेंट से भी कहूंगा कि बीच में प्लीज हैंड रेज ना करें, इससे डिस्ट्रैक्शन होता है जो हमारे स्पीकर होते हैं. हम लास्ट में डाउट क्लियरिंग सेशन कराएंगे. सर काइंडली आप कंटिन्यू करिए.
Okay, thank you. So at stage 2, we will select around 20 from 40 crosses. We can see how much stringent selection we put onto it. And those 20 will be nominated to the AICRP system from different AICRP centers and also the state agriculture universities. So this is about the variety, how we breed a Tiquisite for the varieties. And when we come to the hybrids, as we know, hybrid in earlier our hybrids, we know there are around 2 hybrids which are commercially released. And now there are around another 2 hybrids which we brought into the market of early maturity with the Indian Institute of Pulse Research, Kanpur. So currently, if we take what are the challenges in our earlier hybrid PGNP system is that CMS breakdown in the seed production plots, then hybrid duration, which was around more than 200 days. Then if it is the crop is more than 200 days, we need to have irrigation facility required to sustain the hybrid beyond more than 180 days. And highly photosensitive and thermosensitive parents and hybrid. So the seed production has been restricted to only the rainy season because of its photo and thermosensitive nature. Then also it involves high cost for hybrid seed production because you have to produce in the rainy season and keep it until the next year. So the cost for keeping that seed also incurred in this one, and mainly the restriction is only for that particular rainy season. Then hybrid seed purity testing also issues because we can take only one season per year. Then hybrid seed storage cost, as I told, then less net return to the private seed industry, because if the private seed industry get more money, then only they will take interest in promoting the hybrids. So if you see the heterosis percent and the yield advantage over the cultivated varieties currently, so almost you can see 124% in some of the OFTs in Odisha and some around 47% yield increase and 42% over the best commercial check when you take the hybrids. But some of the hybrid, what happened because of these all problems, they should not move into the large area cultivation in the former field. So recently, because of that, we had a brainstorming session on a hybrid PGNP at ICRISAT with the funding support from Government of India. And currently, government is also supporting a project on developing early maturing PGNP varieties for the specifically dal purpose varieties. So mainly the key recommendation from that particular brainstorming session is that we'll be focusing on the developing a A2 and A4 cytoplasm and early maturing hybrids. That is the maturity of less than 140 days for South, Central and Northeastern plain zones. Then we also plan to define the key seed traits and restore parents for efficient hybrid seed production traits. Then ICRISAT will lead the parental development program in collaboration with Indian Institute of Pulse Research. Then develop parental lines adapted to post-rainy and summer irrigated conditions. Then, but is for the horizontal expansion. Then also the multi-environmental testing through AICRP centers in South, Central and NEPZ zones. Then again, when we do the new PGNP hybrids, the FLD of AVT to hybrid in target zones for at least 1 year before its commercial release, so that their adaptability can be checked before it goes to the commercialization. Then the developed hybrid seed production strategies should be shared with the private seed company so that they can take care of the hybrid seed production and commercial cultivation will be done. So another major important thing with the hybrid PGNP is what we have done is we have classified because the hybrid needs the maintainer program and also the restore program separately. Maintainer program means that bringing up new A lines, and the restore program is bringing up the new R lines. You can see we have a A4 cytoplasmic system, that is the ICP ICRISAT PGNP A line 2039, restorer line with the red color and the maintainer line with the blue color. So when we had a cross with elite lines at ICRISAT breeding program, so you can see they have into different clusters of maintainer and restorer, and some maintainers mixed with the restorer. So what we did is we classify them, and restorers have been identified and put into different clusters so that we can properly develop better heterosis hybrid using the different diversity clusters selection of the material. So these are the 2039 maintainers. We are in different clusters again, around 4 to 5 clusters. When you come to seed and restorer parents, as we know, PGNP is the self-pollinated, often cross-pollinated crop, one of the often cross-pollinated crop among the pulses. And also it has the crossability percentage varies from different varieties to varieties. When we compared for the hybrid breeding program, specifically hybrid breeding needs a open pollinated line, because if as much as cross-pollination happens, that much can be benefited to the farming community, the seed companies and also the farming communities, because the cross-pollination can improve its seed setting percentage. So the seed parent traits in PGNP like CMS lines, we need a stable CMS that is already achieved. Then also we need a photo insensitivity. Then also we need heat tolerance in that one. Then non-seed shriveling nature. Then wilt resistance, SMD resistance, and also it should be having good general combinability, and also having high seed yield as a female parent. Then you should have cold tolerant if the company want to produce in the post-rainy season. And it has to be an open flower. You can see the first two pictures here, one with the stigma exertion, where it can achieve maximum open pollination. And second one is where you can have complete open of the flower so that it can enhance the open pollination and also the crossability of the particular line. So we also need to have a plant height restricted to around 100 to 130 centimeter, then annuality conditions. You can see the dog pigeon pea lines, which is just around 90 to 100 centimeter, wherein it can, if you convert into female line, it can go for open pollination and gives high crossability and produce more hybrid seed. So that is the one required trait for the female parent. Then when we come to the female as well, again female, we need to have photoinsensitive nature. Then heat tolerant, non-seed shriveling, wilt resistance, assembly resistant, high yielding, and good GCA should be there. Then it should have a open flower condition. Because of its open flower, it can move the pollen very easily. Then we need to have a high annuality condition. Then also have high pollen production. Then non-determinate growth habit should be achieved in the male parent so that the continuous pollen production should be achieved. Then plant height of around 130 to 150. In female, we are pushing it to 100 to 90 to 100, but in male, we are pushing from 130 to 150. So that the pollination can be very easy to produce the hybrids. So then when we have the male parent and female parent character, we also have a proper breeding schemes to develop your female parent. That is the TMS sterility conversion. That how we do at ICRISAT actually. So here you can see we use a different kind of scheme to develop those particular A lines and also the R line. Specifically, this is the one where we are developing the seed parent. That is the A lines. We are using this particular kind of breeding scheme actually. We'll be seeing the best B by lines and then we'll be crossing them. That is the elite B into elite B. Then we'll be making around 15 crosses. And from the 15 crosses, we'll be screening the material of around 3-7. And you can see here, since we need to bring a particular line which is having photo and thermo insensitive in nature, you can see them specifically we are dealing with in year 1, year 2, year 3, all the years it is taking under rainy and summer. Because we are pushing the seed production to summer, so that's why we are selecting the material in summer, specifically adopted varieties where the companies can invest their time to the seed increase can be done in post-rainy. This is the one new innovation we are bringing in Pigeon Pea actually. So before going to the conversion actually, the lines which we will be doing with around 15 crosses, around 3750 progenies from F2, we'll be screening for different trait like photo insensitivity, pods per plant, then seed shriveling. Then when we go to the around FI conditions, just we do B by B cross, then before going to conversion, we screen for flowering, wilt assembly, yield, and HSW, that is 100 seed weight. So all the master trait has been captured before we are going to convert it into as a female. Then once it going to female, simultaneously it will go to BC1, BC2. Even in BC2 itself, we do a first test cross with one particular restorer. Then based on that, we are using 80 lines here in the BC1 F1. From that 80, it will be reduced to around 25 with the first TC1. That is called as early generation testing one. Then once we do the EGT1 and select 25, then we go with around 4 to 5 restorers. Again, we do a second TC2, that is test cross 2, wherein we will be selecting maximum 10 best female lines, which can go for the seed increase and hybrid making. And it almost take to bring 10 particular these lines, it will take almost around 6 years of time in PMP breeding to achieve this particular target actually. So if we once we achieve this one, then the hybrid system can go very easily here. So again, same with restorer development also. Since we, as I told, we wanted to do with the summer seed production and also increase the hybrid production and productivity in India. So we are breeding restorer. Restorer also we do a very minimum cross of around 15 crosses here. Then we go for selection. Only in the restorer differences there we are using the conversion program, but here no need to conversion. But again, with the female lines, we do a TC1, TC2. Then we develop best 10 combiners which can give a very good high yield and seed, and also it has a maximum pollen production, which is required for the male line development. That is, then it will go for the seed increase and hybrid making, and it takes almost 5 years actually. The female takes 6 years and the male takes 5 years, but simultaneously those will be running within the breeding system with 2 separate breeders will be handling this one. So again, this is about the breeding scheme which you use at ICSAT breeding program. But when we come to the phenotyping of the trait, which I told, like must-have traits, like Fusarium stulti mosaic virus, and also we studied the trait genetics, then phenotyping and molecular marker. You can see these are the left-hand side, we have a trait, and the middle, we have the trait genetics, and on the right-hand side, we have the phenotyping protocol available, and at what stage we are doing with the molecular markers if we are using in the current breeding program. So next I will show how we use this particular must-have traits to be used in our breeding schemes and how we are selecting the material. These are the first left-hand picture. You can see the rapid generation advancement facility available at ICSAT. Then also we do a field advancement for NA and post-NA for the male and female parent development program, then crossing program, then preliminary yield trials and the other yield trials we are going to take at ICSAT campus. So this is the one, as I told, the wilt and SMT is having a must-have trait. You can see here, ICSAT has developed a historical SIG plot wherein you can have the line screen. Almost we screen around 5,000 lines per year, and almost we reach now the 5 to 10,000 lines. So those 10,000 lines will be screened based on the entire complete breeding program, and we select. Here you can see the line which is having resistant to wilt and SMD, one full line which is from starting to end, that is the resistant line. And the progenies within these 2 rows, you can see the susceptibility and resistant cultivars. So the very stringent screening protocol has been developed, and we see like the lines which we use are highly resistant to wilt and sulti mosaic diseases. Then also we do a off-season SMD nursery at ICSAT Hyderabad, wherein you can screen your material at least 2 times a year. Then also we do if there are lines which are more than 10,000 lines, then we go for control screening for wilt and SMD developed at ICSAT, and we use the different strains like different strains from different states to screen our material and see if the line can move to the different states with that particular resistance. And these are the new lines which we identified in early maturity group, which is having highly resistant to wilt and sulti mosaic diseases. So if we can take around 4 years of our breeding program screening the material. So from 21, we were screened around 1230 lines, and now we are screening almost around 3,200 lines in different maturity groups. And based on the different durations, we are screening different kind of material according to those breeding schemes, which I show in my earlier slides. And another innovation we are bringing with the high throughput phenotyping platform is we are developing a image-based analysis, where you can just push your lines to a image analysis that will show whether susceptible or tolerant line at the early stage of the plant growth. So that we are just standardizing the protocol now. Then we are also standardizing the pod borer. We have not standardized, we have already standardized the artificial diet and rearing protocol for continuous rearing of that Maruca vitrata. This is also one of the new pest which is coming to pigeon pea, specifically the DT type of plants which is most infected. So this is also the first time in the world we are bringing this particular protocol, and that soon will be published. And currently we are using in breeding program screening for Maruca vitrata. The major challenge, as I told earlier, the waterlogging it become a major challenge, and that we have developed a protocol at ICSAT. Specifically, we do 2 types of screening here. One is at the seedling stage, that is 20 days after sowing, because that is the stage where if you get more water, that is affected. And another is the 40 to 45 days after sowing. Those 2 stages are very important. There the maximum rainfall happens in pigeon pea cropping duration and takes maximum damage into the crop. So here we are doing like in 20 days after sowing screening, we do around 72 hours of water logging, that is maximum of 4 days. Then 40 to 45 days after sowing, we are doing 8 days of water logging at ICSAT campus. And the right picture, you see the controlled and also the waterlogging condition. And down here, you can see the resistant varieties for waterlogging. Almost 99% of the lines which are waterlogging susceptible, but very few lines, maximum around 5 lines, we got resistant, and also across the replications, they got highly resistant to waterlogging conditions. The traits which we are considering in our breeding program, like what are the traits which are important is like percent plant survivability, then flowering, whether once we have waterlogging survivability, whether they flower or no. Then once they have the flowering happens, whether they convert into pods or no. Those 3 important traits we are taking, and these are the donors we identified recently from our study for the waterlogging tolerance. Then for Phytophthora blight, actually Phytophthora blight, because the water after waterlogging we have the Phytophthora blight that also causes major damage to the crop. This waterlogging and Phytophthora blight, they come together. Because of the waterlogging only, it will induces the Phytophthora infestation to the crop, and which is also causing major damage to the crop in the current scenario. And major challenge, we need to have the Phytophthora resistant varieties. So Ecrisat, we developed the protocol in 2018. And we have screened almost most of the breeding material, and we got these lines which are resistant to waterlogging, and also some are highly resistant to Phytophthora in this one. The next one is if you want to breed the varieties for the heat tolerance actually. So we have developed a protocol for heat tolerance that is specifically at Ecrisat, specifically planting them in the mid of the January where the temperatures start increasing in the southern part of India. We plant that one and coincide with April, May, very hot conditions up to 42, 43 degrees centigrade. From last 3 years of our experiment, we have identified these are the some best heat tolerant lines, wherein we will be taking around 4 major observations: days to flowering, pollen fertility percentage, pod conversion, and seed sorting. So these most important traits, when we have all those traits for the heat tolerant, then we consider that particular line as a heat tolerant. Then for bringing early maturing varieties, we are using one more important aspect is the growing degree days concept. And we standardize specific days to flowering and days to maturity GDDs. And in each state, we test whether this particular GDD works or no. Then based on that GDD, we select whether this particular line is having photosensitivity or thermosensitivity. So see that these are the different growing degree days in rainy, post-rainy, and summer conditions for the early to late maturing growths. Then for seeds revealing in Pigeon Pea, we are using a specific. We have developed a protocol recently. Within this year itself, we have standardized this protocol from last 3 years study. So in that specifically, we classified the seeds revealing into 5 parts. Actually, class 1 to class 5. Class 5 is highly shriveled. Class 1 is the most non-shriveled one. And also we brought recently a particular machinery that is specifically classified that material into shriveled, into non-shriveled, and give us the percentage which line is good and which line is bad. And this has been developed in Japan, and currently we are using this particular technique or the machinery to breed our material which is non-shriveling in post-rainy and summer conditions. Then finally we came to capping up maturity, that is annuality. As I told, Pigeon Pea is one of the crop where we have the annuality nature is the major challenge. Annuality is less and perenniality is more. So we need to bring that one. If we bring annuality, it will become uniform in maturity. Then it is amenable to mechanical harvesting. Then we will get a very minimum yield loss because the synchronous maturity will be achieved with the annuality. So you can see after screening around 10,000 lines, we got one particular line which is having annuality in nature. The line you can see which is completely maturing, but the line which is just besides still it is growing continuously. So this is the line we identified from our germplasm source and currently introducing into cultivars. And we also brought a line which is having synchronous maturity, annuality, and mechanical harvesting. You can see some of the pictures here. And also we studied the root system of this particular annual line where the seeds root system is dying once it's completed life cycle, but in others it is continuously active. So that is the major challenge we are taking in pigeon pea breeding to bring the varieties which are having annuality in nature. so that the farmers can take particular variety within a particular period of time, then go for the next season. Then we also develop a protocol for the for the pod borer, because this is also one of the major challenge. At least 50-60% of the pod borer resistance can be achieved with our varieties with this particular protocols. Then lastly, when we call about vegetable pigeon pea, so you can see when we can call a particular variety as the vegetable pigeon pea, this protocol we have standardizing, and currently we are at this particular stage. You can see if you want to call particular line as a vegetable pigeon pea and when to harvest, because the farmer has to get his benefit for the vegetable pigeon pea, so that you can tell when you start flowering, when you can harvest and deliver it to the market as a vegetable pigeon pea. So here, based on the flowering nature and also the pod formation and also the grain formation within that pod. So if you go to the pod formation stage of around P21 to P22, and the seed formation stage of S20 to S21. That means days after flowering. That 16 to 17 days after flowering, you can harvest and say this can go for the vegetable actually. So that means that is the green pod stage available, and which is we can have taste of that one. And after the S21 stage, it will automatically start becoming a grain. And within 6-7 days, it's complete its life cycle and become a matured grain. So you can also see here differences within the grain as well. The premature seeds, vegetable pigeon pea, over mature seeds, and the dry mature. You can realize the vegetable pigeon pea is having highly green and also fully filled seeds. And in premature seeds, although it is green but not fully matured, so you will not realize the taste of that one. And in over mature seeds, it will be little bit start making yellowish color. Then the taste will become reduced. And another important thing in vegetable pigeon pea, we have the TSS, that is soluble sugar, will be more during the vegetable pigeon pea stage compared to the previous the first one and the latest two stages. So this is the vegetable pigeon pea variation we found in our breeding material, starting from 3 seed to 7 seed. Then last portion of our presentation is how we are using the genomic resources and phenotyping platforms currently available at ICRISAT. Because in the recent years, we need to reduce the time to take breeding. So we are currently at ICRISAT, as I told, we have the full sequence genome available for the crop. And that is reference genome available with around more than 90% precision. Then we have a whole genome resequence lines and pan genome work is in progress. Then we have a gene expression atlas for all the pigeon pea available, and we can study each gene from this particular expression atlas. Then for the genotyping purposes, we have the high density array of 54K. Then we have mid density array of around 2000K. Then low density of around 40 markers. Then also we have trait associated markers available in our ICRISAT, so that they can be used in enhancing the efficiency of selection in the breeding cycle. So apart from that, we are also implementing the genomic selection model development. Currently, we are working on 4 aspects here. One is the multi-location and multi-environmental phenotyping of the stage 1 trials, where we are screening around 200 entries in around more than 110 locations across India. And after that, we are also genotyping those particular entries in the multi-environmental testing. Then we'll be implementing a G by E trait association, keeping in the mind of G by E interaction of the genotypes into environment. Then finally, we do this, develop the GS model and select the material for the, some goes to the, as a parent to our recycling parent, and also goes to the next level of testing. So with the genomic selection, we can reduce still more time compared to the current breeding cycle actually. Then according to our GS model development, we also done a static TPE analysis, that is a target product population of environments, where if you take India, actually, India in almost 400 districts, the pigeon pea grows, but only 291 districts where the major production comes. So we are identifying what are those major production zones and how the varieties adapt to there actually. Then that can give us particularly the breeding of new pigeon pea varieties and enhance our yield by taking those particular genotypes which are having high yield advantage over the TPEs. And the yellow one font label in this one is ICRISAT will do a multi-environmental testing with the NAS partners across India from Punjab to Tamil Nadu. So these are the soil parameters we taken from different locations for our multi-environmental testing location for the genomic selection model development. So that the each and every aspect of the soil and the microbial flora and also the environmental characterization will be covered so that our model will be very most reliable and do a breeding program quickly with the GS and develop new material. So these are some of the trialing across India we have done last 2 years for bringing new early maturing pigeon varieties. So finally, we have this rapid generation protocol that specifically I told the pigeon pea is a crop where it takes only 1 season per year. And for that, it used to need around 1 to 7 years of breeding, 2 to 4 years of testing, and another 1 to 3 years of release. Almost 12 to 13 years to the release time to bring a new variety. So in this optimization of photoperiod regime in pigeon pea, we selected different genotypes that is having early, mid-early, medium, and late. Then we have the genotypes with genotypes and also the treatment of around 8 hour, 9 hour, and 13 hours of photoperiod and temperatures which is required for that one. For the proper crop growth stage and humidity around 60 to 70% and nutrients required for the crop. And we have done this in 4 chambers using these particular light and intensities and different light intensities and genotype. You can see from A to H different stages of crop growth and different light intensities we are using to produce in rapid generation advancement. So if we take the matrix of speed breeding, you can see the early maturing, we can take up to 4 generations per year. In mid-early, we can take 3 generations per year. And in medium and long duration, we can take at least 2 generations per year. And this particular protocol has been published in Springer method, nature plant methods, so that you can open access, you can also take a look into this particular. And we also have the speed breeding protocol setup that can save up to 45 to 60 days early. The one big advantage of this particular method is you can see in the natural condition, we can take only 1 generation per year, but within the speed breeding, we can take around 4 generations per year. And another cost per plant also, you can see if you use the cost per plant, single cost is almost $1.33 in field and also $1.3 in RGA. But field is restricted to only time bound, but the RGA is not time bound. And if you use the measure, the cost involved is the electricity consumption. If we can reduce the electricity consumption, then it can reduce half of that cost which you use in the field. And there are some high chances of losing the breeding material because of the natural climatic weather, but that is not there in the RGA where you can 100% assure. And here with developing new variety takes almost 12 to 13 years, but with RGA we can take around 4 to 6 years for breeding new varieties. And we also brought a seed chip genotyping protocol method, wherein you can just take part of that seed chip and go for the genotyping and predict its particular genotype, what is there in the that particular line before going to plant. That is, we are bringing the precision in this one. Then also recently we brought a new pigeon pea varieties into the heat tolerance and mechanical harvesting, where you can go for the very short height with a high machineries can be used to go for the mechanical harvesting, so that it will be very helpful for the farmers to reduce the time taken for the crop. And if you grow this particular crop in the summer condition, almost it can reduce the duration, and also it can give higher the yield compared to the rainy season. So you can see the left hand side, the machinery once it harvests, it will give the complete harvest within a very short period of time, so that the labor consumption is also very less and the farmers can get more benefit to the crop. So finally, we ICRISAT in collaboration with the national programs, we are working to develop bring new varieties into the system to help with the farmers. Recently, we also organized a annual group meet of the Kharif Pulses 2024 at ICRISAT. Then also organized a capacity building for the NOS program Then we developed the world's first PGNP bulletin on world first PGNP protocol. And we also done a capacity building of the people. Almost we train more than 100 people, 100 persons which work on this particular crop from different parts of the world. Then these are the partners and they are donors to our particular breeding program. And with this one, these are some of the new innovative techniques which we use in current breeding program to enhance the genetic gain. And this particular thank you slide, you can see the lines which you brought, which can come within 90 days, can be grown in the rice fallow environments actually. So with this, I will conclude my presentation. Thank you for hearing my talk.
एडमिन प्लीज चैट बॉक्स ऑन कर दीजिए आप किसी भी पार्टिसिपेंट्स की यदि कोई क्वेरी है इस पर्टिकुलर टॉपिक को लेके आप डिस्कस कर सकते हैं चैट बॉक्स में आप लिखिए उस क्वेरी को सर तक पहुंचाया जाएगा और सर उसका रिप्लाई करेंगे रोहित जी क्वेश्चन आया है पढ़िए उसको
जी सर. So sir, we have first question. In North Hill zone, huge demand is caused by pod borer. Huge damage is caused by pod borer complex, especially Mora species and Helpicorva species. Are there any varieties which is available or used against these insects, sir?
Currently in breeding program, actually we have some variety, but which are not 100% resistant. We can say around 50-60% resistant. Along with this, you can use the natural measures or the any chemical which can control the pod borer.
Thank you, sir. Then we have next question that what is the ideal growing season and how does it fix nitrogen by pigeon pea, sir?
The ideal growing season in India is the rainy season, starting from June to, we can plant up to second week of July. Then harvest by January or the maximum by February actually. And it fixes nitrogen based on its root nodulation actually. It is a leguminous crop. So natural rhizobium causes the root nodulation to fix its nitrogen. And almost around 60 to 80 kg per hectare it can fixes within its cropping duration. The peak rhizobium or the nitrogen fixing stage is the after around 30 days after planting, and it will start decaying once it starts the flowering condition. And as it completes its life cycle, then the complete decay of the rhizobium will happens. But that particular nitrogen peaks by during that cropping season, which will help to the next crop where it can take the nitrogen from the soil.
Thank you, sir. Then we have next question. Is Rhizocotonia cause foliar disease in pigeon pea, sir?
Rhizoctonia.
Rhizocotonia, यहाँ से लिखा था ये Rhizocotonia. I don't know if I'm doing this. Might be some spelling issue, sir. So okay, so we have next question, sir. That can, sir, which variety of pigeon pea will be good and also disease resistance for Eastern Uttar Pradesh?
We have now IPA 15-6 that is highly resistant to wilt and SMD and adopted to Western Uttar Pradesh.
Okay, and actually, sir, we are getting lots of questions regarding variety. Next question is, sir, please suggest some short duration and of pigeon pea variety, sir.
That IPA 15-6 is short duration only. And we have one more ICPL 87, that is a DORP variety.
Okay, the next question is, sir, in UP region, sir, like during maturity of pigeon pea, there is not uniform. So can you suggest some variety which has like uniform maturity of pods?
Yeah, all early maturing varieties which we are bringing now, they have uniform maturity. So they will come like we are promoting currently ICPV 21333, that is having uniform maturity actually.
Thank you, sir. Then we have next question, sir. Can pigeon pea is intercropped with another crop, sir?
Yeah, majorly in Maharashtra, it is almost 80% is intercropped with sorghum. It is intercropped with cotton. It can be intercropped with maize.
Thank you, sir. Then the next question we have, like, is there any work or research currently being conducted on biofortified pigeon pea variety, sir?
Hmm?
Biofortified pigeon varieties, like, so, like, is some work is going on that thing, sir, or not?
Yeah, we are working on calcium and magnesium, but still it is in the progress. Not at the moment we are bringing directly into the market as a biofortified product.
Okay, thank you, sir. And we're getting also greetings, sir. Nice presentation, good presentation, sir. Like very informative session. And I think we have one more question. Sir, can you please explain about the control of flower drop in pigeon pea?
Control of flower drop, actually in pigeon pea we are using the pulse magic actually. If we can, there is a one particular product developed from University of Agriculture Science, Raichur. So this can be sprayed during the peak flowering period, and it can enhances the productivity of almost around 10 to 15% through not to drop more flowers.
Thank you, sir. And we have, I think, done all the questions. Ankur, sir,
जी
Thank you.
thank you, sir.
सारे क्वेश्चंस का जवाब मिल गया है। बहुत बहुत धन्यवाद सर कि आपने बहुत ही बढ़िया सेल्फ एक्सप्लेनेटरी लेक्चर दिया और मेरी रिक्वेस्ट पे आपने ज्वाइन किया सर। थैंक यू सो मच। सभी पार्टिसिपेंट्स लेफ्ट कर सकते हैं यदि उनकी कोई क्वेरी नहीं है। बहुत बहुत धन्यवाद प्रकाश सर।
Thank you, Ankur ji.
Thank you.
I can
Thank you,
leave
sir.
now.
यस सर प्लीज।
Okay. Thank you, ji.
Thank you, sir. Thank you.